Okay, so today we are starting another topic. In the last video we were done with formation of three germ layers. Today we are going with events of neurolation. So let's see. I hope you'll enjoy and understand nicely. So when it comes about this term, you should always be none. So let's see what this neurolation means. This simply means formation of brain and spinal cord. Formation of brain and spinal cord. So I'm sure it would be clear. Then comes even we just consider these are neurulation, which is derived from what? Derived from ectoderm, which is the outer germ layer. Ectoderm. So what will happen after ectoderm? This is derived from nervous. When we just consider about nervous, so this means if I just tell you definition, it will be this one, or if we just say differentiate of ectoderm from brain to spinal cord. So differentiation of ectoderm from brain and spinal cord, from brain and spinal cord. So I'm sure it will be clear. So if we just look into examples, you'll understand it much nicely. So okay, here comes the example. Let's see. If we just talk about organogenesis, this will be our next topic hopefully. But as we just have gotten term for examples, organogenesis is simply formation of organs, organ formation. So what you'll see, organogenesis, which means organ formation. When organ will be formed, then we'll consider, of course, if a baby is going to be born, so we need brain and spinal cord. Even these are the first steps which we made, brain and spinal cord. So we just consider them as a neurulation. So I'm sure it will be clear. Neurulation is simply making a brain and spinal cord, and hopefully it will be clear. Then when we just talk about the mechanism, so I would love to introduce that as well. So here come mechanism. Let's go with this color. Okay, mechanism. So what I'll just talk about, let's see. First one, you'll see ectoderm as this the starting ectoderm. After this ectoderm, you'll see another ectoderm thickening. After ectoderm thickening, there would be neural plate. After this neural plate, you'll see neural growth. I'm just introducing these all, rest we'll see in. I'm just making a picture and a flow chart, deepen, which then we'll see neural fold. After this neural fold, we'll see superior margin fusion. And then we'll see neural tube. Neural tube is quite good topic. Well, after this neural tube, we'll see they are again divided into two parts, which are brain and spinal cord. This is finally brain and spinal cord will be formed. So I'm sure it won't be very much clear to you. So let's see what's written in the book, what we see next. Then we'll introduce neural tube and neural crest cells, migration, these all. Okay. Let's see. So here we can see these pictures. So let's see. So we are discussing neurolation, events of neurolation, which mean the steps which I mentioned. Then neurolation is simply by the process which neural plate forms and eventually become the neural tube. So first thing we'll see neural plate. When they eventually become neural tube, when they will rise to the brain and spinal cord during the third week of development. So we have so many stages of development, which I'll just make you understand in neural tube, which we'll discuss a few minutes later on. Then this one during the third week of development, ectoderm thicken the middle, forming neural plate. Neural plate will be formed. Neural plate which begins to invaginate or fold inwards, forming the neutral. So they'll become fold-like structure that's called a neural growth that runs along the middle of the embryo. Then the neural plate continue to invaginate and they form neural folds, which this kind of folds we'll see with the picture. Then on neural on either side neural groove okay start to elevate and approach such until they form the neural tube. Then as neural tube is forming some of the cells, they will form neural crest. Then neural crest will eventually rise to the various cell types including cranial and spinal lump. So this means this combined they form the structure as the same cellular level organization. We know atom forms cell, then cell combined they form tissue combined they form organ, then organ system, then complete living organism. So this all, so as I mentioned, these are the flowchart. They will eventually fold these all things. We'll see with the picture, but before I just continue, let's see detail about neural tube, as that's mentioned and quite important topic. So neural tube is simply we have two parts. How? If this the picture, first one you'll see would be anterior, second one you see called as posterior. Okay, posterior. So this means in posterior we have spinal cord. This means at the back side. When it comes to front side, that's called as a brain, right? So spinal cord and these all, then this brain is again divided into three parts. We know this divided into forebrain, the midbrain, and the hindbrain. So I'm sure it will be clear to you these three. So what? How will we see this? Simply, this all things will be seen in the first month. So the three structure will be formed. When we talk about the second month, what will happen? The brain fluxes. This means brain fluxes. Fluxes simply mean kind of not opening, but the folding, which we can see. Folding, of course, we know brain structure is quite complicated and have something like these random, like this one. These structures, that folding can be seen in the second month, right? Then comes the third part, which is about what the third month. That's making brain waves. Not the third month, but that's even happened the second month. So let's see. We'll see brain waves as well, which can be seen. Curve. We can see the bending. These all foldings will be also seen. So as mentioned, that everything done in the third week, and here we have reached to the second month, just because in the third week, like first month or the third week, there's a formation of the first more important part. Of course, before making that organs, we are supposed to make, not we, but as generally this is a very thing that we need to know. Brain is formed for the very first, so that other functions can be analyzed by the brain, as brain is the control system like CPU and everything we might have heard in a childhood, like. So I'm sure it will be clear then these things. So let's see, here till here it was clear. Then comes this region, which I believe is kind of blur. If I just add them color. Okay, let's see. We'll see diagram later on. Then, for example, anterior and the neural tube, which form the brain, while the posterior and which form the spinal cord, as I mentioned. Then the neural tube eventually close at the both end with the cranial head, and yes, again this come neural crest cell basically. which are here, but here they've been introduced. So neural tube eventually grows at the both end, while the cranial head and closing first, followed by the caudal tail. So of course we know head would be the start and ending would be done on the tail. Then we have neural crest cell, which is the downward. So let's introduce them, but what I believe we'll do that in the next video. But for now, let's see this diagram, these folds and everything. So let me just add the picture again so that it won't be very much clear. Okay, so I've directly opened with the PDF. Here you can see the structure. First thing, we have neural plate and the border. Border is simply the middle green line, and below that, like outside that, that's we have an outer membrane called as ectoderm. The middle one called as neutral plate, right? And then after neutral plate, we have the outer, which is mesoderm, which is simply the second layer. Then the middle, you see notochord, right? After this, what will happen, they will convergence. This means they form a round like a structure where neutral crest is formed, and this structure for become a folded like structure. So neurus crest cell, which is also component of PNS, which is periphery nervous system. Then there's ectoderm, which will make a kind of fold here in the middle. Then when the fold is there, then the middle neural tube is formed. You can see like that structure. Then after that, we have these all things, which is epidermis, the outer one. Then we have notochord, which is mesoderm derived, and the neural tube, which is this tube internal. Here we have green structure, which are called as signal ganglion. And yes, and here these are mesoderm derived. So yes, I am sure to be clear. Thank you for watching. Then we'll come up with this video of neural crest and neural cells. Thank you for watching. Bye bye.